get started. To get a clinical perspective, let's look at a quick flash case. Let's say a mother calls the clinic for a telehealth visit because her 10-year-old son has been experiencing cough, a runny nose, headaches, and fatigue for the last 5 days. She states it's similar to the illness her child experienced 2 months ago. The patient is known to the practice and he has a history of recurrent viral infections. Let's keep this in mind as we go through the lecture. Our learning objectives are to differentiate natural killer cells from other cellular components of the innate immune system, and to recognize the functions of natural killer cells. Okay, to start things off, let's recall that a natural killer cell is a lymphocyte member of the innate immune system, and let's stop right there for a second. What is a lymphocyte? Remember, lymphocytes refer to B cells, T cells, and natural killer or NK cells. B cells and T cells mediate our adaptive immunity, while NK cells are part of the innate immune response. Here we see a microscopic view of lymphocytes. Recognize that they have a very round, densely staining nucleus, pointed out by this blue arrow, with a small amount of pale cytoplasm pointed out on the edge with this red arrow. Okay, now that we've discussed lymphocytes, let's talk about the innate immune system. Like we just reviewed on the last slide, T cells and B cells are part of our adaptive immunity, while natural killer cells are part of our innate immunity. Remember, our adaptive immunity is highly specific, which develops memory over long periods, compared to our innate immunity, which is nonspecific, has no memory response, and occurs rapidly. So, what are our natural killer cells in our innate immunity nonspecifically and rapidly acting on, and what are they doing? Well, naturally, they're killing. But what are they killing, and how? Natural killer cells use perforin and granzymes to induce apoptosis of virally infected cells and tumor cells. So let's take a look at this diagram of a natural killer cell. Remember, it's a lymphocyte with a big, round, densely staining nucleus, but in the cytoplasm, we will find some lytic granules. These lytic granules contain perforin and granzyme, which act on target cells, like virally infected cells and tumor cells, to induce apoptosis. The activity of natural killer cells is enhanced by IL-2, IL-12, interferon alpha, and interferon beta. Natural killer cells are induced to kill when exposed to two different things. The first would be to a nonspecific activation signal on a target cell, or to an absence of an inhibitory signal such as MHC class I on a target cell surface. Okay, so what would cause a nonspecific activation signal on a target cell? What would cause a cell to have certain cell surface proteins that say, hey, natural killer cell, come over here and tell me to undergo apoptosis? If you're thinking of some sort of stress, infection, or injury, then you're on the right track. A natural killer cell can distinguish between healthy and infected cells by identifying cell surface proteins that are induced by stress, malignant transformation, or microbial infections. Okay, and the other exposure in which NK cells would be induced to kill would be to an absence of an inhibitory signal, such as MHC class I on the target cell surface. Now, why wouldn't it have an inhibitory signal? Well, because the cell is trying to say something's wrong and saying it should undergo apoptosis. A good example would be a virally infected cell, in which those molecules are downregulated. Lastly, natural killer cells also kill via antibody-dependent cell-mediated cytotoxicity, sometimes abbreviated ADCC. Can you recall what type of hypersensitivity this falls under? Yep, you're right if you're saying type 2 hypersensitivity. On an NK cell, CD16 on its surface will bind to the Fc region of IgG on the abnormal cell, activating the NK cell. Now, this is depicted well in this image, where the NK cell is the green one on top, and the abnormal cell is at the bottom. Now, in the middle, we can see the blue CD16 on the NK cell binding with the Fc receptor for IgG on the abnormal cell. Okay, that was a lot of information. Let's do a quick quiz. When are natural killer cells induced to kill? NK cells are induced to kill when exposed to a non-specific activation signal on a target cell and or to an absence of an inhibitory signal. Let's go back to our flash case. So here we have a boy with recurrent viral infections. Now, there are a number of immunodeficiencies, but as we just discussed, natural killer cells play a vital role in killing virally infected cells. While a natural killer cell deficiency is rare, it is something that can be considered in a child with recurrent viral infections. So what's the bottom line? Remember that natural killer cells are lymphocyte members of the innate immune system. They use perforin and granzymes to induce apoptosis of virally infected cells and tumor cells. Their activity is enhanced by IL-2, IL-12, interferon alpha, and interferon beta, and they are induced to kill when exposed to a non-specific activation signal on a target cell and or to an absence of an inhibitory signal. And they also kill antibody-dependent cell-mediated cytotoxicity. In short, those are the functions of natural killer cells. That's all I have for you here. Please give this video a thumbs up if you liked it, and it'll give you the opportunity to submit a comment if you have any feedback or questions. Thanks for joining me. Study hard.
Hi everyone, this is our lecture on the major functions of B and T cells. So let's get started. To get a clinical perspective, let's think that there is a 2-year-old boy who's brought to his pediatrician by his parents because he has a fever and sore throat, which he also experienced last year. He was seen 3 months ago with impetigo. Despite recurrent infection since approximately 6 months of age, on physical exam, there is no apparent lymphadenopathy. His tonsils are small. What is the most likely diagnosis? So, our learning objectives for this lecture are to recognize the major functions of B and T cells and to compare and contrast major immune mechanisms of B and T cells. Okay, so let's talk about the major functions of B and T cells, and let's do B cells first. B cells are a major component of our humoral immunity. What is humoral immunity? Well, back in the day, the immune system got divided up into humoral immunity and cell-mediated immunity. And cell-mediated makes sense. It has to do with some of our main immune cells, like our T cells, and when microbes get inside our cells. If cell-mediated is so focused on cells, then why did it leave out the B cells? Well, humoral immunity got its own category because its focus is on stuff outside of cells. So components that are in our fluids, like our antibodies and complement proteins, and our B cells lead the way. What are our major functions of our B cells? Well, they're super important because they recognize antigen and they're capable of presenting that antigen. Remember, they are antigen-presenting cells. Also, they undergo somatic hypermutation to optimize their specificity for antigens. You should recall this image. When an antigen is encountered at the antigen binding site, then we can mix up the variable region to generate antibodies that are specific to that antigen. Isn't that cool? So now we can recognize and present the antigen. We got a little ahead of ourselves and we know that B cells can also produce antibody. How do they do that? Well, the B cells can differentiate into plasma cells, and then they can secrete specific immunoglobulins. Now, if we encounter that same antigen again, thanks to our B cells, we can maintain immunologic memory because memory B cells persist and can be faster at responding to the same antigen when it meets it again. Up next, we have our T cells. If B cells are primarily our components considered in humoral immunity, T cells are part of our cell-mediated immunity. Now, we do have different types of T cells. Our main two types are our CD4 positive T cells and our CD8 positive T cells. Our CD4 positive T cells are also called our helper T cells, and that's because they help out our B cells to make those antibodies. They even produce cytokines to recruit phagocytes and activate our other leukocytes. That sounds super helpful, right? Our CD8 positive T cells are also called our cytotoxic T cells, and that's because they directly kill virus-infected and tumor cells with perforin and granzymes, like our natural killer cells. What's perforin again? Yep, you're right if you said it perforates. It makes holes in target cells, and then granzymes come along and induce programmed cell death, or apoptosis in that target cell. T cells also play a major role in our delayed cell-mediated hypersensitivity, which is type 4. Can you name an example of a type 4 hypersensitivity? You're right if you're saying poison ivy or some sort of contact dermatitis or a graft versus host disease. Speaking of graft versus host disease, that leads us to another major function of our T cells, which is in acute and chronic cellular organ rejection. Lastly, our T cells have major histocompatibility complexes, or MHCs, on them, and they can be remembered with the rule of 8. MHC2 is on CD4 cells because 2 times 4 is 8, and MHC1 is on CD8 cells because 1 times 8 is 8. All right, time for a flash quiz. What cells are part of our humoral immunity and produce antibodies? You're right if you said B cells. Okay, one more. Which type of T cells directly kill virus-infected and tumor cells? You're right if you said CD8 positive or cytotoxic T cells. So let's get back to this kid. What's his problem? He keeps getting a fever and sore throat, and he's had impetigo before too. He's only 2 years old. Seems like he's having problems eliminating bacteria. His lymph nodes aren't enlarged, and his tonsils are small, which doesn't make sense, because our B cells that help us with bacterial infections should be proliferating in our lymph node follicles and germinal centers. This kid likely has a B cell disorder, like X-linked or Bruton's agammaglobulinemia. Without B cells maturing, he's not going to have a lot of antibodies produced, and it is a typical finding to have decreased immunoglobulins of all classes with this immunodeficiency. So our bottom line for this lecture can be summed up like this. The major functions of B cells include humoral immunity, such as recognizing and presenting encountered antigens, producing antibody, and maintaining immunologic memory. The major functions of T cells are cell-mediated and include CD4 positive T cells, helping B cells to make antibodies and producing cytokines, and CD8 positive T cells directly killing virus-infected and tumor cells and being involved in delayed hypersensitivity and acute and chronic organ rejection. Well, that's all I have for you here. Please give this video a thumbs up if you liked it, and it'll give you the opportunity to submit a comment if you have any feedback or questions. Thanks for joining me. Study hard.
It is important to understand the process of how T cells differentiate and where the particular differentiation process occurs within the thymus. T cell precursors are produced in the bone marrow and are soon selected via positive and negative selection in the thymus. Positive and negative selection are two quality control mechanisms for your T cell production. They make sure that you don't end up with T cells that overreact to normal healthy cells in your body. The first precursor cells move on to the thymic cortex, where positive selection occurs. This first test is for all cells with a toll cell receptor to see if they can make an antigenic reaction to self-MHC. This step is basically asking, do you work? If no response is witnessed, you're out. The cell undergoes apoptosis. Only those worthy and functioning properly get to move further through the gauntlet. Challenge number 2, negative selection. This step takes place in the thymic medulla and is essentially testing whether the TCR response is too strong. So, negative selection essentially asks, do you work too well? We are interested in antigenic overstimulation because that is a mechanism by which autoimmune diseases arise. The T cells get too trigger happy and destroy their own healthy tissue. The ones that do not pass either apoptose or become regulatory cells. The surviving T cell precursors differentiate into either CD4 positive or CD8 positive mature T cells. At this point, the CD8 positive T cells migrate to the lymph node where they are presented with antigen. CD4 positive T cells, or helper T cells, are further divided into different subcategories: Th1, Th2, Th17, and T regulatory cells. IL-12 stimulates the CD4 to differentiate into T helper 1 cells. Th1 cells release cytokines that stimulate B cells to change which type of antibodies they produce, such as IgG instead of IgM. They also stimulate macrophages to consume and degrade pathogens. IL-4 triggers CD4 cells to develop into T helper 2 cells, whose main function is to release cytokines that cause B cells to start producing IgE antibodies. These rest on the surface of mast cells until an antigen triggers the release of inflammatory allergic cytokines from mast cells. TGF-beta induces CD4 T cells to differentiate into both T helper 17 cells and Treg cells, which help with neutrophilic induction and immune cell regulation, respectively. Flash quiz. Although both arise from the bone marrow, where do B and T cells mature? Bone marrow for B cells and thymus for T cells. B and B, T and T. Macrophage-lymphocyte interaction. When a lymphocyte binds to a macrophage, it releases interferon gamma, which enhances macrophage phagocytic activity and recruitment. It is essentially like giving the macrophage a high-octane energy shot, and this gets it really excited to go and phagocytize foreign particles. This, in turn, creates a cascade of events that helps to quickly bring on an immune response. This interaction is enhanced by CD40-CD40 ligand binding. Cytotoxic T cells. Cytotoxic CD8 T cells are responsible for the destruction of infected, neoplastic, or otherwise disordered cells. They kill these cells by releasing proteins that puncture the cell membrane as well as activate the apoptotic cascade. This is very similar to what we see with NK cells. They have CD8 on their surfaces, which binds to the MHC1s of infected cells. And since CD4 cells secrete interferon gamma, it would make sense that this would dampen the CD8 cells. You'll see a wide variety of autoimmune diseases in first aid that all wreak havoc on multiple organ systems, sometimes simultaneously due to misdirection of the body's normal immune response. Regulatory T cells help keep this from happening by suppressing unwanted immune activity. They do this by suppressing the CD4 and CD8 T cell functions via anti-inflammatory cytokines like IL-10 and TGF-beta. They also express the receptors CD3, CD4, and CD25. Now, since these cells are crucial in maintaining control of our immune response, it stands to reason that an absence of these cells would lead to some sort of autoimmune pathology, and it is called IPEX, or Immune Dysregulation Polyendocrinopathy, Enteropathy, X-linked syndrome. Yeah, I prefer IPEX too. This syndrome is a result of a deficiency of the FOXP3 gene, which is crucial for autoimmunity. A patient with this would present with GI symptoms, nail dystrophy, endocrine issues, and other autoimmune disorders. It is also associated with diabetes in male infants.
Hi everyone, this is our lecture on T and B cell activation, so let's get started. To get a clinical perspective, let's look at a quick flash case. Let's say a father brings his 5-year-old son to the clinic because he's sick again. The patient was previously ill due to infections with pneumocystis and CMV. The father states he has had watery diarrhea for the past 4 days, and no one else in the household has similar symptoms. A stool sample is collected, and a few days later, the results reveal cryptosporidium. Let's keep this in mind as we go through the lecture. Our learning objectives are to summarize the roles of T cells and B cells, to enumerate the steps of T cell and B cell activation. Well, before we dive into activation, let's recall some of the major functions of B cells and T cells. Remember that B cells are part of our humoral immunity. They recognize and present antigen and undergo somatic hypermutation, optimize their specificity to an antigen, leading to producing antibodies and maintaining immunologic memory. T cells, on the other hand, are part of our cell-mediated immunity. Our CD4 positive T cells help the B cells out by making antibodies and producing cytokines, while our CD8 positive T cells directly kill with perforin and granzyme. Those are pretty important roles, so we want to make sure they're activated to do those things. Another few bits of info we need before getting to activation. Let's recall that we have a few types of antigen-presenting cells. including B cells, dendritic cells, Langerhans cells, and macrophages. Additionally, something to keep in mind for this lecture is that two signals are required for T cell activation, B cell activation, and class switching. Okay, let's start with T cell activation. The first step in this process is that an antigen-presenting cell, again, so it could be a B cell, a dendritic cell, Langerhans cell, or macrophage, has to ingest and process an antigen, like in this image. But then it really doesn't know what to do with that antigen, and it's going to go looking around for help. And it goes to a lymph node, which is where it encounters a naive T cell. Now the next step involves our first signal for T cell activation, and it depends on whether or not the antigen is exogenous or endogenous. If it's an exogenous antigen, it is presented on MHC class II, like it is here, and recognized by a T cell receptor or TCR on the helper T cell or CD4 positive cell. And if it is an endogenous or cross-presented antigen, then it is presented on an MHC class I on TC on a CD8 positive T cell. Now the next step is that second signal we need for activation, and it's about proliferation and survival. A co-stimulatory signal occurs via interaction of the B7 protein or CD8086 on the antigen-presenting cell and CD28 on the naive T cell. Lastly, now we have an activated helper T cell, which can produce cytokines, while a cytotoxic T cell will be able to recognize and kill a virus-infected cell. Okay, now let's go through B cell activation and class switching. The first step will be helper T cell activation, like we saw in the last slide, which we can see right here. After that, we then have B cell receptor-mediated endocytosis, where, as you can see in this image, the B cell receptor on the surface of the B cell, it gets that antigen because it's one of our antigen-presenting cells, and it brings it into the cell. Next, that exogenous antigen is presented, and it's recognized by the T cell receptor on the helper T cell. Now, since it's a helper T cell or CD4 positive T cell, can you remember which type of MHC is doing the presenting of that antigen on the B cell? You're right if you're thinking it's an MHC class II, which is what we can see happening in the image here. Great, so we've got one signal. What comes next? The next step will be the CD40 receptor on the B cell, which we can see here, binding to the CD40 ligand on the helper T cell. After that, the next step will be the helper T cell secreting cytokines that determine the immunoglobulin class switching of the B cells. And lastly, the B cells are activated and produce IgM. They can then undergo class switching and affinity maturation. Well, that was a lot. Let's pause for a second and do a quick quiz. What are the two signals required for T cell activation? Remember, for T cell activation, we need antigen presentation by the APC and recognition by the T cell, and a co-stimulatory signal from the B7 protein, CD8086, and CD28 on the T cell. Okay, let's get back to our case. We have a 5-year-old boy who is sick again. The patient was previously ill due to infections with pneumocystis and CMV. Father states he's had watery diarrhea for the past 4 days, and no one else in the household has similar symptoms. A stool sample is collected, and a few days later, the results reveal cryptosporidium. Now, even kids who get sick a lot usually aren't coming down with pneumocystis, CMV, and crypto. Now, there are a number of immunodeficiencies, but one in particular leads to these types of opportunistic infections, and that's hyper IgM syndrome. As we discussed, the helper T cell will secrete cytokines that determine the immunoglobulin class switching of B cells. If this doesn't happen, then we'll just have a lot of IgM. Can you remember this step in B cell activation and class switching right before the helper T cell gets to secrete those cytokines? You're right if you're thinking of the second signal, which is an interaction between CD40 and CD40 ligand. Now, hyper-IgM syndrome is an X-linked recessive disorder, and it's most commonly due to defective CD40 ligand on the helper T cells. Because of this, patients will have very, very low levels of IgG, IgA, and IgE because they aren't getting that second signal for activation and class switching. So, what's the bottom line? Remember that two signals are required for T cell activation, B cell activation, and class switching. In T cell activation, there's antigen ingestion, antigen presentation and recognition, a co-stimulatory signal, and activation of that helper T cell or cytotoxic T cell. In B cell activation and class switching, we start off with helper T cell activation, then antigen ingestion, antigen presentation and recognition, co-stimulatory signal, secretion of cytokines, activation and production of IgM, and class switching and affinity maturation. In short, that's T and B cell activation. That's all I have for you here. Please give this video a thumbs up if you liked it, and it'll give you the opportunity to submit a comment if you have any feedback or questions. Thanks for joining me. Study hard.
Hi everyone. This is our lecture on antibody structure and function, so let's get started. To put some clinical perspective to this lecture, let's say a 23-year-old man presents to the office with complaint of abdominal cramping, nausea, bloating, and diarrhea. He recently returned from a hiking trip with his friend. They all drank water from streams, but he is the only one from his group experiencing symptoms. What is a likely diagnosis? Our learning objectives for this lecture are to recognize the structural components of an antibody, associate antibody structure with function, differentiate antibody diversity from antibody specificity. What do we need antibodies for anyway? They help our immunity, but how do they function? Well, let's talk about what they do first. Well, we know that they help us recognize antigens. And when they do, they can participate in neutralizing an offending agent. The antibodies, which are these individual structures we see in the circle, look like the letter Y, and they can surround a microbe and prevent its adherence. What else can our antibodies do? Well, it wouldn't be helpful if we had all these bacterial particles neutralized and just persistently floating around. So our antibodies also help with opsonization, which promotes phagocytosis. Now, I like to think of opsonization as using a highlighter. Amidst all that text in your notes, when you highlight just one thing on the page, it stands out. It's kind of what opsonins do. It helps emphasize what needs to be phagocytosed and makes the whole process more efficient. Anyway, antibodies also help with complement activation. Antibodies can activate complement and enhance opsonization and lysis. So, keep in mind, we need our antibodies to recognize different antigens so that they can be neutralized, and then we need to help get rid of the bad stuff with phagocytosis and complement activation. Okay, let's talk about the structure of an antibody and how it helps us with our antibody functions. It looks like the letter Y. So first of all, we have two pairs of chains. Here is the first set, made up of these grayish heavy chains, and here is the second set, made up of these green light chains. Now the heavy chains are connected at this bendy central hinge region by disulfide bonds. The light chains are connected to the heavy chains by disulfide bonds too. This bottom part is known as the FC region, and you can see it only has the gray chains or heavy chains in it. This top part of the letter Y is known as the Fab region, and you can see it has both gray heavy chains and green light chains in it. Okay, now that we know the details of the structure, let's talk about each big region one at a time. Let's start off with the Fab region, the arms of the Y on top, right here with this arrow. The whole point of this part of the antibody is for the recognition of antigens. Fab actually stands for the fragment of the antibody in charge of antigen binding. So fragment F, A for antigen, and B for binding. So that's a great way to remember it. If we look more closely at the Fab region, we see on the diagram that there are variable and hypervariable regions at the tips of the Y. Why do these have to be variable or different? You're right if you're thinking that there are so many different antigens out there, and our antibodies need to be made specifically to interact with them. The Fab region does include both the gray and the green parts, so both heavy and light chains. Ultimately, this Fab region is what determines idiotype. The idiotype is the characteristic of the antibody that makes it unique for an antigen, based on the variability of the Fab region. In fact, only one antigenic specificity is expressed per B cell. Now that we've discussed the Fab region, let's move on to the FC region down below. It's the stem or the bottom of the Y, pointed out by this arrow. Well, what does the C stand for? Well, you can think of it as the constant region of the antibody that has carboxy terminals. It is also where complement binding occurs. In this region of the antibody, there are also carbohydrate side chains. This is the part of the antibody that confers or determines what isotype the antibody is. So, IgG, IgM, IgD, and so on. Well, now that we're more familiar with the details of functions and structure of antibodies, how do we make them so diverse? We actually have random recombination of genes that you see in the variable regions. So we have these blue arrows pointing to V and J regions on the light chains, and red arrows pointing to V, D, and J regions on the heavy chains, and these can undergo recombination. We also have the random addition of nucleotides to DNA during the recombination process. And this is accomplished by the enzyme terminal deoxynucleotidal transferase, or TDT. Finally, we can also generate antibody diversity with random combination of our heavy chains and our light chains. Well, if our antibodies can be so diverse, how do we know how to make them specific? Then you have to ask, specific for what? Well, an antigen, of course. So generating this is an antigen-dependent process. This occurs through somatic hypermutation and affinity maturation in our variable region, and isotype switching in our constant region. Great! Now that we've covered all that, let's check what we know. Does complement bind on the Fab region or FC region? Great! You're right if you said that complement binds on the FC region. Well, let's get back to this case then. When we think of a camper or a hiker, especially if we know they had water from a stream, we're likely thinking of what protozoa? Yep, you're right if you're saying Giardia from the cysts in the water. But how come he's the only one experiencing symptoms? Can you recall which immunodeficiency increases an individual's susceptibility to Giardia infections? Yep, you're right if you're thinking selective IgA deficiency. Remember, IgA is produced in the GI tract and protects against gut infections. IgA prevents the attachment of bacteria and viruses to mucous membranes through neutralization. It can recognize all the different antigens because of the variability of the Fab regions. So, in summary, remember that antibody structure is highly related to its function, especially for neutralization, opsonization, and complement activation. Antibody diversity is generated independent of antigens, and antibody specificity is generated dependent on antigens that are presented by somatic hypermutation, affinity maturation, and isotype switching. Well, that's all I have for you here. Please give this video a thumbs up if you liked it, and it'll give you the opportunity to submit a comment if you have any feedback or questions. Thanks for joining me. Study hard. Hi everyone. This is our lecture on immunoglobulin isotypes. So let's get started. Let's start with a case. A 10-month-old boy is brought to his pediatrician's office by his grandmother because he seems as if he's sick again. The boy was seen 1 month prior with an ear infection and 2 months prior due to fever and vomiting. His grandmother asks why he is suddenly sick all the time when he was doing well in his first 6 months of life. How do we explain this one? Our learning objectives for this lecture are to define affinity and avidity, then to recognize the main features of the immunoglobulin isotypes, differentiate the immunoglobulin isotypes based on structure and function, and to associate clinical scenarios with predominant immunoglobulin isotypes. Now, before we dive into the individual isotypes, let's talk about them in general. Now, all isotypes can exist as monomers, so even though we talk about some of them being dimers or pentamers, they can all exist as monomers, or single units. Now, remember, where do we get our immunoglobulins? Remember, they're produced by our B cells that differentiated into plasma cells, which secreted our immunoglobulins. Now, we've heard of B cell maturation and how they go from stem cell to pro-B to pre-B and so on. But what we end up with in the maturation process are mature, naive B cells. And why are they naive? They're naive because they haven't had any exposure to an antigen before, which means prior to activation. Well, those mature but naive B cells express both IgM and IgD on their surface. If they encounter an antigen, then these B cells may differentiate in the germinal centers of lymph nodes by isotype switching into plasma cells that secrete the other immunoglobulin isotypes, IgA, IgE, or IgG. How do they know what to switch to? Our immune system loves sending messages through cytokines and ligands like CD40 ligand, and these will induce gene rearrangement to make the appropriate isotype. Now, there are 2 words we need to get familiar with because they get mixed up a lot, and they come up a lot in questions. So when the word affinity is used, they're referring to an antibody and antigen interacting with each other, but just one antibody and one antigen. So here's an antibody, and here's an antigen binding to the Fab region. And when we talk about affinity, we're talking about this individual interaction. When we talk about avidity, we're talking about the cumulative binding strength of all antigen-antibody interactions in a multivalent molecule. So here we have a multivalent molecule, or a dimer, because there are 2 components. And we have an antibody and antigen interaction over here, and over here, and over here, and over here. And the cumulative or sum total of all of these interactions is avidity. So let's start it off with IgG. IgG is our main antibody in our secondary response to an antigen, and it is the most abundant isotype in our serum. It also takes part in fixing complement. Remember, what activates the classical complement pathway? Antigen and antibody complexes with IgG or IgM. IgG also can opsonize bacteria and neutralize bacterial toxins and viruses. While all of these things are great, what makes IgG really unique is that IgG is the only isotype that crosses the placenta. Why is this a big deal? Well, it provides infants with passive immunity. We like to use the mnemonic IgG greets the growing fetus, but its levels wane after birth. Next up is IgA. IgA prevents the attachment of bacteria and viruses to mucous membranes. We know it does not fix complement. When we talk about IgA in circulation, it exists as a monomer, but when it is secreted, it has the J chain like you see in the image below, which makes it a dimer. IgA crosses epithelial cells by transcytosis, and it is produced in the GI tract and protects against gut infections. This is an important correlate because remember, selective IgA deficiency is the most common primary immunodeficiency. And since IgA protects against gut infections, these patients have an increased susceptibility to Giardia. Well, what else should we know? IgA is the most produced antibody overall, but it has lower serum concentration. It is released into secretions like our tears, saliva, mucus, and breast milk. It picks up secretory components from epithelial cells, which protects the Fc portion of the antibody from luminal proteases that would otherwise destroy it. Next, we have IgM. It is produced in the primary, immediate response to an antibody, and it fixes complement. It serves as a receptor for antigens on the surface of B cells. But when it is on a B cell, it's a monomer. When it is secreted, it's a pentamer. Just like IgA, it needs the J chain to link more than one unit together. What's so cool about this pentamer? Well, it enables avid binding to antigen while humoral response evolves. What's avidity again? It's the cumulative response of all of these antibody and antigen interactions. Next, we have IgD. And honestly, its function is still unclear, but it's found on the surface of many B cells and in the serum. Lastly, we have IgE. IgE binds mast cells and basophils. It cross-links when exposed to allergen, so it mediates our immediate type 1 hypersensitivity through release of inflammatory mediators such as histamine. It is also important because it contributes to immunity to parasites by activating eosinophils. I know that was a lot of information, so let's do a few quiz questions. What is the only immunoglobulin isotype that crosses the placenta? Yep, it's IgG. Which immunoglobulin isotype is secreted as a pentamer? Yep, it's IgM. Which immunoglobulin isotype is secreted into tears, saliva, mucus, and breast milk? Yep, it's IgA. So let's get back to this case. Why does this boy seem to be so sick all the time? He seems to be having recurrent bacterial and perhaps enteroviral infections, especially with the vomiting. He was fine his first 6 months. Why is that? Well, which antibody do we know that crosses the placenta? Yep, it's IgG. And remember, its levels start to wane after birth. If those were the only antibodies this boy was depending on, when those levels really get low, like at 6 months, then he's going to be really prone to infections. But why would he have low immunoglobulins of all classes? There could be a number of reasons, but a common immunodeficiency is X-linked or Bruton's A-gamma globulinemia. And A-gamma globulinemia really means without gamma globulins, and emia means low levels. In this disorder, there are absent B cells in the peripheral blood. And remember, we need our B cells to differentiate into plasma cells and produce all the classes of antibodies we just discussed. So what's the bottom line? There are 5 immunoglobulin isotypes: IgA, IgG, IgD, IgM, and IgE. And they are different in structure and function and are predominant in various clinical scenarios. Well, that's all I have for you here. Please give this video a thumbs up if you liked it, and it'll give you the opportunity to submit a comment if you have any feedback or questions. Thanks for joining me. Study hard. Hi everyone, this is the lecture on antigen type and memory. Let's get started. So antigens are either thymus-dependent or thymus-independent, and we can categorize them this way based on the involvement of the cells that mature in the thymus, which are the T cells. So let me tell you a story, which I hope will help you better understand this concept. So let's say you're doing rounds at the hospital, and a patient who is wearing a gown is in trouble. And you get a page from your chief resident, your attending, and the nurse on call. Well, you're going to run without thinking and help out. All patients on the floor are wearing gowns, but because there were so many repeating signals to you, you went directly to the patient on your own, pretty independently. Now, let's say you're doing rounds, and a patient has decided that his gown was way too boring, and he's decided he needs to decorate his gown with stickers, or dog stickers. You notice the patient, because not all patients have dog stickers on them. You notice that another resident is particularly fond of dogs. So you go to that resident and say, hey, look, that patient over there, he has dog stickers on his gown. And that resident says, wow, no way. And you've caught the resident's attention. And it turns out that the resident's favorite candy bar is the one he sees sticking out of your white coat pocket. So now together, you and the other resident work on that patient that day. So which patient are you more likely to remember? I think it's the one with the dog stickers. So in this example, the patient is the antigen. And you're an antigen-presenting cell, so let's say you're a B cell. When you want to show your resident the patient, you're acting like MHC, and your resident is a T cell. And the candy bar is acting as another T cell and B cell interaction, perhaps the CD40 and CD40 ligand. So, okay, let's talk about thymus-dependent antigens first. Now, thymus-dependent antigens, they have a protein component. It's similar to the stickers in our story. This allows presentation of the antigen via MHC. When these antigens are presented to the appropriate cells, they induce a rapid and robust response that is specific to the antigen. Now, that interaction between an antigen-presenting cell like a B cell and a T cell allows class switching and immunologic memory to occur. Now, here's an image of B cell activation and class switching involving a thymus-dependent antigen. Now, in real immunology, we don't have stickers, but we do have protein components. And some examples are the Streptococcus pneumonia PCV13 vaccine. And we also have polysaccharides that can't get presented to T cells on their own, so they are conjugated to diphtheria toxin-like protein. Now, thymus-independent antigens, on the other hand, they don't have a peptide component. like the lipopolysaccharides from gram-negative bacteria. So because of this, they can't be presented by MHC to T cells. They still create an immunologic response, but it's very weakly immunogenic, and it doesn't lead to memory. These antigens are only able to stimulate the release of IgM. The vaccines based on this concept, this often requires boosters and adjuvants, just like the capsular polysaccharide subunit of the Streptococcus pneumonia PPSV23 vaccine. So I hope the story helped you understand thymus-independent and thymus-dependent antigens. That's all I have for you in this video. Thanks for joining me. If you have any feedback or questions, please click on the thumb icon and submit your comments or questions there. Thanks. Hi everyone, this is our lecture on complement, so let's get started. The complement cascade is a series of proteins that complement the rest of the immune system and help the inflammatory response. Now, what organ makes a lot of proteins, like our plasma proteins and clotting factors and carrier proteins? You're right if you said the liver, and the proteins of the complement cascade are made there too. Now, complement plays a role in our innate immunity. Can you remember some characteristics of innate immunity? It's like our reflexes. We were born with them, and we were born with complement. If something gets in my eye, I'm going to blink. I don't care if it's snow, rain, dirt, or volcanic ash. I'm going to blink. If something bothers my immune system and gets in my way, my innate immunity is going to kick in. It doesn't really care too much what it is either. It's non-specific, and it happens very rapidly, like in minutes to hours. And part of our innate immunity is complement. Now, complement also plays a role in inflammation. Why is that? Well, inflammation, especially acute inflammation, is our early response to injury or infection. Acute inflammation is actually a reaction of our innate immune system, so it makes sense that it's also characterized by what type of cells. Yep, those are neutrophils in the tissue. I know that all these cascades in medicine can seem very overwhelming, like the coagulation cascade, but what you need to focus on are how these pathways get stimulated and what happens when part of the complement cascade is deficient or not working. Now, the coolest complement protein is C3b. Everyone wants to get C3b's attention. Why is that? Well, once C3b figures out what's going on, it can activate C5 convertase and ultimately get C5b, C6, C7, C8, and C9 to form MAC, or the membrane attack complex. And what happens when a cell membrane gets attacked or damaged? The cell can lyse. So like I said before, everyone wants C3b's attention. Who's everyone? Well, there's the classic pathway, the lectin pathway, and the alternative pathway. And as you can see, they all lead to C3b. Now, the classic pathway activates complement with antibody and antigen complexes. The lectin pathway is activated with microbial surfaces, with mannose as an example. Sometimes this pathway is called the MBL pathway or the mannose binding lectin pathway. And lastly, the alternative pathway can be activated spontaneously or by microbial surfaces, and it amplifies the generation of our cool cat, C3b. So, as you can see, there are 3 pathways, but they all converge at C3b. So we can make the membrane attack complex to help in our innate defense against gram-negative bacteria. One very high-yield point to know is that deficiency of any part of complement can lead to increased infections. But if we're missing any part of the membrane attack complex, we are extremely susceptible to recurrent Neisseria infections. Now, like I previously mentioned, the classic pathway activates complement with antibody and antigen complexes. The antibodies that bind to the antigens are IgG or IgM. One way to remember that is that GM makes classic cars. Just as a recap of the other two, the alternative pathway gets triggered by microbe surface molecules, and the lectin pathway is activated by mannose or other sugars found on the surface of the microbe. So let's talk about some of the components of the complement cascade and their functions. These are extremely high yield, so make sure you know these. Which one should we talk about first? You're right, we should totally start with C3b, since it's so central to all the pathways. C3b is important for opsonization, and it also helps to clear immune complexes. So what does opsonization mean? Opsonin is actually Greek, and it means to prepare for eating. And when I think of cells eating in microbiology and immunology, I think of one cell engulfing a part of something, or maybe a whole something, but that's phagocytosis. The two primary opsonins when we're talking about our body's defense against bacteria are C3b and IgG. One way to remember this is to remember that C3b binds to lipopolysaccharides on bacteria. There are a lot of B's in there. Now, C3a, C4a, and C5a are also very important components of complement, and they all have A in it. It's a great way to remember that these three play a role in anaphylaxis. Now, let's talk about some more complement components. What about C5a? C5a plays a role in neutrophil chemotaxis. What does this mean? Chemo means chemical and taxis means to move. So why does there need to be movement of neutrophils somewhere? Just for some recall, I've added in this image. And this is the story of how leukocytes, or more specifically neutrophils, as shown in the image as PMNs, exit from blood vessels to get to sites of tissue injury and inflammation. First, they marginate to the edges of the vessel, and they roll along. Second, they go through tight binding or adhesion. Third, they go through endothelial cells and exit the blood vessel in a process called diapedesis or transmigration. And finally, they migrate through the interstitium to the site of injury or infection. But how do they know where to go? Well, that's what chemotactic factors are for. They hold up a sign and say, hey, neutrophils, there's inflammation because something's going on and we need you over here. Can you name any additional chemotactic factors you may have previously learned? You're right if you said IL-8, leukotriene B4, kallikrein, and platelet activating factor. And let's not forget about C5b, C6, C7, C8, and C9, which together cause cell lysis when they form the membrane attack complex. Now, there are also inhibitors of complement. Why would we want to inhibit complement when it's part of our innate immunity? Well, we don't want it to get out of hand. We like things to be regulated. Our inhibitors help to prevent complement to activate on our own cells. Two important inhibitors are decay accelerating factor, also known as CD55, and C1 esterase inhibitor. You can imagine if one of these two inhibitors were deficient, we may run into some issues. C1 esterase inhibitor deficiency is associated with hereditary angioedema, and decay accelerating factor deficiency is associated with paroxysmal nocturnal hemoglobinuria. All right, that's it. Thanks for joining me. Please give this video a thumbs up if you liked it, and it will give you the opportunity to submit a comment if you have any feedback or questions. Thanks again. Study hard.